Great. Well, thank you very much for the introduction. Also, thanks for the invitation. It's really an honor to kick off the third day of what's been an instructive and inspiring summit and symposium. So thanks to HFSP and all the staff for putting this together. So you all know from life that unlike you and I, plants don't slather on SPF 50 when they go outside on a sunny day. Yet, except in a few limiting cases, they don't get burned. Now, the reason for that is because they've evolved their own version of sunscreen. One of the major focuses in my lab is understanding the molecular mechanisms behind this sunscreen. The reason that we're so interested in this process is both to advance fundamental knowledge and because of what's shown here. So I showed this 2 days ago, but I think it bears repeating. What we see here is for 4 major cash crops, yield as a function of time, with predicted demand shown in dotted lines and predicted supply in dashed ones. And we see this growing mismatch between supply and demand. And so one of the biggest challenges facing us as a society, as Beverly laid out, is figuring out how to bridge this gap. One way to do it is to increase the supply. So fundamentally, the supply comes from photosynthesis. But we know from our fundamental studies of photosynthesis that it operates at this measly 1 to 5% power conversion efficiency, meaning just this small percentage of energy gets used to generate the biomass of the plant, including all the food that we eat. And we know that the rest of the energy goes to a range of different processes: carbohydrate synthesis and inefficiencies in that reaction, the metabolic processes like photorespiration, and environmental responses, which I've drawn as the biggest slice of this pie here, which is, for example, the conversion of absorbed sunlight to heat. But the size of these slices changes with factors like nutrients and weather. And so if we really want to understand what controls the size of these slices and begin to think about ways in which we can change them, we need to look at molecular mechanism. So looking at a crop field like this one, we want to zoom in on the molecular machinery. And we find incredibly complex nanomachinery. The protein shown here, I introduced briefly at the summit, but it's protein near and dear to my heart. It's called light harvesting complex 2. This is the protein that makes leaves green. It's also the most abundant membrane protein on Earth. And so this protein is responsible for capturing sunlight and in part controlling the partitioning of that sunlight between the different slices of this pie. So if we want to understand these slices, and today I'm going to focus on one of them, thinking about how absorbed sunlight generates heat, one of the things that we know is that this level of heat can change dramatically, like I laid out at the beginning, for example, with weather. So I showed it originally at 70% of absorbed energy gets converted to heat, and that can certainly happen on a sunny day. But under clouds, it can be less, taking on a range of different levels. So if we want to think about optimizing the amplitude of this slice of pie, what we need to think about is what are the molecular mechanisms at play in this protein here. So if we want to think about the molecular mechanisms of dissipation, we can start by thinking about why is it that dissipation is happening at all? Why is it that these proteins have evolved processes to take sunlight and convert it to heat? And that's actually shown in this plot right here. This shows sunlight on a typical day at a typical point on the Earth's surface. And what we can see is a profile of sunrise, sunset, along with clouds and shadows. Now, sunlight up until around this level is useful. This matches the turnover rate of the molecular machinery of photosynthesis. But typically energy in excess of this is dangerous. A little more rigorously, what happens is the chlorophyll molecules involved in photosynthesis absorb sunlight, undergo what's called an intersystem crossing to generate what's known as a triplet state that can sensitize singlet oxygen or reactive oxygen species. And these reactive oxygen species, as all the biologists in the room know, damage cellular machinery. In fact, they're why I hope you all eat fruits and vegetables, because these reactive oxygen species or free radicals can damage our own machinery, and we need the photoprotective mechanisms of plants to eat them to keep them safe. So this is what they've evolved to do in plants. We can't do the same thing, so we eat our fruits and vegetables to make sure. All right, so getting back to this, so ideally, plants would dissipate the excess energy, retaining only the useful, but all of the useful energy. Now, the problem is that plants are cautious. They keep much less than this ideal level. And so if we want to think about this mechanism, what we fundamentally need to understand is how is it that this level of dissipation is controlled. So this is a regulatory process, which means we can conceive of it as a control loop, where we have our input, our desired output, and then the control loop that converts that input into the output and sets that level of that dissipation process. And so the fundamental question, if we want to think about this slice of pie, is what are the molecular mechanisms behind this control loop? And so what we know is that energy gets regulated via a protein conformational switch. So proteins, like light harvesting complex 2, have embedded within them two types of pigments: chlorophyll, the primary pigment of photosynthesis, and carotenoids, which serve a variety of roles, including as the free radical scavengers that we need to eat. And so it's these chlorophyll and carotenoid molecules that capture and manage solar energy. So they can absorb sunlight and use it to direct it towards the generation of biomass, or they can redirect that energy into heat. So we want to understand the conformational or structural switch of this protein that goes between these two states. So one of the mechanistic questions about dissipation is, what is the conformational switch that turns it on? And so this is the mechanistic question that I'm going to talk to you about today, as an example of the kind of work that we do in my lab to understand dissipation in plants. Okay, so what's been known for decades from a range of molecular biology and biochemistry studies is that the level of dissipation gets activated by two factors: by a drop in pH, which means there's accumulation of protons, and a change in the chemical composition of one of the carotenoids, where it gets converted through an enzyme from violazanthin to zeaxanthin through a deepoxidase reaction. And I'll just say that that activity of the enzyme is in turn controlled by pH. So it's known that the dissipation, or the conformational switch, gets activated by these two factors, but what changes in this protein had been a long-standing question. And one challenge is, if we want to understand the conformation of the protein, typically when we do protein-based experiments, biochemical or spectroscopic, we might be measuring around 10 to the 4th individual proteins. So each one of those proteins will have the switch toggled to a different state, meaning it's in a different conformation, or undergoing a different part of the dynamical process. And so typically what happens is measurements average over all of these things. And so one of the things that we do in my lab is we take these proteins and we put them in a really sensitive microscope, so we can look at just one protein at a time. We can identify its conformation to determine the state of the switch. Now, what we actually measure in these experiments is what's called the fluorescence lifetime of the protein. How long it takes sunlight or solar energy to come back out. This tells us about the level of dissipation. If energy is only coming out for a short time, that means there's more dissipation. And so this gives us a way to determine the state of dissipation in individual proteins. From measuring many of these proteins, we can figure out what is the state of the switch under different conditions, putting together a picture that's known as a free energy landscape, which fundamentally tells us about the relative populations of different protein conformations under different conditions. And this gives us a window into the level of dissipation and how it changes. Along with this, we can look at the dynamics to figure out how this process responds to different changes in sunlight. So we've taken this approach and looked at a range of different photosynthetic organisms. We've looked at proteins from green plants, and remarkably, we didn't really see any dynamics. Tells us that the plants get fixed, or these proteins in the plants get fixed in a certain level of dissipation. We looked at moss, and we found slow millisecond and microsecond dynamics. This is slow for a protein. And we looked at green algae, where we saw microsecond dynamics. So one of the things that's interesting about thinking about these processes in different species is that we can compare them to figure out what kind of balance of these different processes gives us the best combination for things like increased biomass. And so from these studies, we were able to determine that some of the conformational switches in plants occurs in an associated protein. So these screens here are not really working. If somebody could fix that, they keep switching on and off. Thanks. All right. Well, we'll kind of wing it. Okay. Along with that, in moss, we can see that there are slower dynamics, which is consistent with the environmental conditions of moss. We know that moss grows in these safe and protected environments, damp and in the shade. And then finally, if we look at the proteins from green algae, we see much faster dynamics. And so today, as an example of our work, I'm going to tell you just about these proteins from green algae. Also because we're interested in thinking about green algae for different biomass production. All right, so I showed you before this free energy landscape associated with the level of dissipation. And so one of the things that these initial experiments showed us is that we were able to identify a protein conformation with dissipation on and with dissipation off. Now, by doing some additional experiments, we were able to find that there's not just a single conformational switch in these proteins, but actually two different conformational switches, where there's two different structural changes to the protein that can turn the level of dissipation on. By a series of other experiments, we found that one of these switches is controlled by the pH drop, and the other of these switches is controlled by the carotenoid composition. This tells us that there's two distinct conformational switches in the protein, one controlled by pH and one controlled by carotenoid composition. By a lot more detailed analysis and experiments, what we're able to do is take this information and map it back onto the protein structure, where we're able to determine where in this protein the likely source of these conformational switches is. So from these spectroscopic experiments, we could put together this mechanistic picture of how these conformational changes occur. Along with this picture, we could do correlative measurements of what's happening in vivo. So we can measure the same parameter in green algae, where we're measuring again what's called the fluorescence lifetime, or for how long energy comes back out. The fluorescence lifetime gets shorter. That tells us that the level of dissipation is increasing. And what we saw in wild type is that dissipation increased under sunny conditions, as you'd expect. And then we looked at a series of mutants. First, we looked at a mutant that only had the pH-dependent dissipation, and we saw that we could recover the effect of dissipation. Then we looked at a mutant that had only the carotenoid dissipation, and we saw that regardless of the light condition, which changes the pH, the level of dissipation was maintained. So this is consistent with the in vitro experiments that show us that there are these two distinct processes to turn on dissipation in vivo. One controlled by pH and one controlled by carotenoids. And so we can conceive of this as the organism having two separate knobs. One where the level of dissipation is regulated through pH, and one where the level of dissipation is regulated through carotenoids. So thinking about this, we now see these two separate knobs responsible for tuning this control loop that I introduced at the beginning that sets the level of dissipation. So that's the picture that our spectroscopic and biochemical experiments were able to determine of dissipation and how it works in the protein. But now you may be wondering, why two? What is the evolutionary advantage to having these two separate control knobs for dissipation? And so to think about that a little bit more in detail, we can think more deeply about this control loop. So fundamentally, this is a regulatory process. If we think about a control loop, like what controls the temperature in this room, it's known as a PID loop. That means there are three separate components: a proportional, an integral, and a differential component. It's how temperature is regulated in rooms and how basically any control system works. And so the proportional component is proportional to the input. The integral component is proportional to the integral of the input. And the differential component is proportional to the differential of that input. Now, what we know from basic control theory is that if you have a fluctuating input, you need the integral control. And if we look at our input here, sunlight on a typical day, what we see is that it fluctuates. I mean, we also know this. We've seen clouds, right? And so that tells us that if we think about how we're going to regulate the level of energy in response to sunlight, we need integral control. So I told you that one of the dissipative conformational switches is controlled by pH. So fundamentally, what is pH? It's the accumulation of protons. So that accumulation of protons gives you a single integral control. So we know from control theory that a single integral control regulates a step change, like we see when a cloud or a shadow passes over something. Now, if we think about the other component, the carotenoid composition, we know from the biochemistry that the carotenoid composition is regulated by an enzyme. So it depends on the accumulation of that enzyme. But the activity of that enzyme is pH-driven. So it depends on the accumulation of pH. So we're single integral control of the enzyme, but double integral control of proteins. And if we again turn back to control theory, a kind of basic loop like we have occurring in this room, this double integral control regulates a ramp change, something like sunrise or the breaking up of cloud cover. So fundamentally, what these studies have told us is that these proteins have evolved to have two separate conformational switches that activate dissipation. And by thinking about this in the context of the environmental responses, what we've been able to see is that these two conformational switches are able to respond to the two types of fluctuations in sunlight. All right. But I motivated all this by thinking about, can we actually increase the amount of biomass? Is there a way to increase the amount of energy? And so if we want to think about how to do that, we need to think about how things change when we turn these knobs, or when the level of dissipation changes. So it's been well established for decades that you can knock out the enzyme responsible for the carotenoid conversion. So that knob, we have established strategies to control. But pH, which is part of the cellular feedback loop generated through photosynthesis, has been much more challenging to turn that associated knob. So to begin to investigate that question, one of the things that we did is went back to this protein and figured out a way to remove the section of it sensitive to pH. And what we were able to see is that removes the component associated with the conformational change, or the conformational switch that activates dissipation. So, in thinking about this conformational switch that responds to types of fluctuations in sunlight, we've been able to identify the presence of these two separate switches that respond to the two types of fluctuations in sunlight. And whereas one of these switches has been well established, that you can control it in the organism, we've begun to show a pathway for a way that you can turn the other of these switches off. So collectively, the ability to turn these two control knobs provides us a platform to test hypotheses of how we would be able to set the switch to the ideal levels for increased biomass. Because if we want to think about ways to adjust the level of the slices of this pie that I introduced in the beginning, so this large component that leads to things like heat can be reallocated as biomass. And so I told you just one story today of one aspect of the mechanisms behind the dissipation of excess energy as heat. This is just one example of how our fundamental knowledge of the complex biochemical and biological reactions of photosynthesis can provide us a pathway to develop a blueprint for the systems and structures associated with photosynthesis that may lead to this reallocation of energy into biomass. All right, and I want to spend my last couple minutes just making sure to acknowledge all of the people in the group that did this work. So there's been a lot of discussion so far about the importance of transdisciplinary work, and I want to start by saying that photosynthesis is inherently a transdisciplinary field where everything from quantum mechanics to agricultural science come together to understand how this works. A lot of the work that I told you about was in collaboration with the plant biologist Roberto Bassi, who's in the audience today, who provides not just protein, but really important biological insight and perspective. Along with Roberto, we've been lucky enough to work with Matteo Ballatari and Alberto Pinola as collaborators and colleagues on this work. And finally, much of what I presented today was developed by a former postdoc in the group, Toru Kondo, who's now an associate professor at Tokyo Tech doing beautiful work in his own group. And of course, I want to thank the funders, and in particular, the Human Frontiers Science Program that not only hosted this, but helped supported some of our more radical ideas about understanding this process. Thank you.